Central to the understanding of cells is what sometimes is referred to as the central dogma in biology, that living cells use DNA as the code for these cells. This code is replicated during cell division, that this DNA code can be transcribed. It can undergo transcription to make an RNA copy of the code, and then the RNA copy then determines the amino acid order of proteins, and then the proteins run the cell. And so this is certainly very important, but there's more to life than that. I'm going to focus on that aspect of cellular mechanisms, but there's more which should be discussed later. So for example, if you were to consider the genome, and let's imagine each one of these red DNA double helices represents 1% of the genome, so the human genome. Then some of it, or sorry, most of it is transcribed, half to more than half. And so half to more than half is converted into transcripts, RNA transcripts in the process of transcription. And this is called the transcriptome. Now, it's a little harder to study because the genome stays permanent in the nuclei of cells. The transcription rates vary. And so different cell types, liver cells are transcribing different portions of the genome to brain cells. One makes different transcripts during embryonic development than adulthood. Cancer cells may make, abnormal, areas transcribed. And so the transcriptome is harder to study, but not all of the genome codes for RNA. Much of it does, half, maybe 60, 70%. So a lot of it is transcribed. What percentage of the genome codes for protein? Well, it ends up, that's a very small percentage, actually. So if you look at the amino acid order of the proteins, maybe 3% of this genome was actually determining the amino acid order of proteins. Now, this is important because the proteins are, including the ion channels, which are conducting electricity in the brain, or the actin and myosin performing contraction in muscle, or the keratin of hair, etc. So these proteins are obviously important, but they are not the majority of the genome. It does not encode these amino acid structures. So I'm now going to then introduce the process of a translation, and in the next video, go through it more fully. But I need to say at the outset, not every portion of the genome is transcribed, and not every portion of the transcriptome is translated into proteins. And so once we discuss translation, we shouldn't assume that that's it. There's a lot of RNAs which have function without ever being converted into protein, and they should be discussed then subsequently. Okay, so this is now going to introduce the genetic code and translation, so that then a lecture can proceed on translation. So just as RNA and DNA are polymers made of lots of little molecules called monomers, proteins are polymers made of lots of joining monomers known as amino acids. Now, there are 20 basic kinds of amino acids. Technically, we can add 2 more, so technically it would be 22, but let's just stick with the basics. There are 20, and so these are the building blocks. So if you think of, say, Legos, for example, you can assemble all of these pieces to make a great variety of different structures, but all of those different structures would then have the same pieces. It's just the order in which they are joined, which determines the overall structure. And so there are these 20 amino acids, and as you put them in order, which ones you use makes a difference, because some are, say, polar, all right, like the ones given here. Some are non-polar, so some mix well with water. Given that the outside of the cell is mostly water, the inside of the cell is mostly water, obviously polar amino acids, which mix with water, they could be used well in those portions. There are portions of the cell which are not polar. They are non-polar, like the interior of the cell membrane where those phospholipids are. So here in orange, these non-polar amino acids would serve. Some amino acids are acidic, some are basic, some have aromatic rings. And so there are these 20 amino acids, and their features vary in important ways. So if you are building a protein, yes, you're going to be joining, different amino acids in a chain, but which ones you choose will then determine the nature of the protein. And so that's what the DNA, which gets transcribed into RNA, has to tell us, which order, right? So, how do we build this protein? And it ends up being, very important, and this can be stressed then later, because as we consider this, for example, here is an enzyme, a digestive enzyme, chymotrypsin. It is called a serine protease, and it is called that because serine is one of 3 vital amino acids which exist in the active site of the enzyme. So a little pocket into which a reactant fits. If you put a different amino acid there, then the active site would have a different shape. The reactant wouldn't fit, and it would not be that digestive enzyme. The amino acid in a protein is so vital that very often mutant proteins, say ones which result in cancer, or here's an androgen receptor, which is going to be mutant and not function. And you ask why? Well, because the normal amino acid leucine was replaced by an arginine. Or why is there cancer? Well, because the normal amino acid proline is replaced by a threonine. Or if here's an enzyme which won't make estrogen, and the way it should, because the normal arginine was replaced by cysteine. And so it is very important to put the right amino acids in the right places, especially not to change, say, a polar amino acid for a non-polar amino acid. And so, as I'll describe in the next videos, then we talk about the structure of proteins in various levels. So before I talk about translation and making a protein, let's just kind of define proteins, what our end goal is. First off, there's what's called the primary structure of proteins. The primary structure of proteins is the amino acid order. Which of those 20 do we put in which order? Once again, because a mistake could then be a mutant protein which does not function and could result in cancer or the like. And so the DNA code is the DNA code here, this double helix, has a nucleotide order. And then the nucleotide order, where each cytosine and each adenine then is encoding information. This is converted into an RNA order. And so here you can see that there are RNA nucleotides in the transcript here in aqua. And then in the process which we're building up to, translation, then that order of RNA nucleotides is read to determine the order of the amino acids which are inserted. Okay. So the primary order of the primary structure of a protein is determined by the order of the amino acids. But proteins don't form these straight filaments. They can fold, they can bend, they can take on various shapes. So the order of the amino acids forms the primary structure, but what happens after that? Well, there are a couple of shapes which can result, and a commonly used one is what's known as an alpha helix. All right, so collagen, for example, has 3 different proteins, 1, 2, and 3, all forming alpha helices. Now, the reason that an alpha helix is such a commonly used motif is if you look here, notice that the amino acids can be arranged so that their side chains, which can have varying sizes, can all face outward. So it's a way of packing the amino acids close together because the small portion of the amino acid is what's forming this helix center, and the larger side chains can be accommodated because they're going outwards. So this is an alpha helix. It's commonly used, so some proteins like collagen are just alpha helices. And in collagen, for example, you could use 1 collagen gene and then use 3 different blue collagen proteins and put them together. But there are different collagen genes. So you could use a blue collagen gene, an orange collagen gene, and a green collagen gene. So there can be different, collagens in the alpha helices here. So an entire protein could be made of alpha helices, but very often a protein has just it's this big structure, and small sections of it could form alpha helices. So the entire protein doesn't have to be an alpha helix. That could just be a portion of the protein. So the alpha helix is a commonly used secondary structure motif. Another commonly used is the beta-pleated sheet. So notice here's this amino acid, where this amino acid, the side chain goes up, the next one, the side chain goes down, then the next one goes up, then down, and up, then down, then up, etc. But now we could fit in a neighboring amino acid chain that could then be reversed, in that where this side chain for this amino acid is going up, then the next one could then be going down. So this one goes up while its neighbor goes down. This one goes down while its neighbor goes up, etc. And once again, this is a way then of packaging these together. Silk is primarily made out of a beta-pleated sheet. So some proteins are primarily beta-pleated sheets, but then there are also proteins where a portion of the protein could be a beta-pleated sheet over here and an alpha helix over here. So those types of things like alpha helices and beta-pleated sheets form the secondary structure of a protein. Then we can form additional levels by taking, say, this part of the protein and attaching it to that part of the protein. This will determine what's called the tertiary structure of the protein. Now, for example, the amino acid cysteine has sulfur atoms in it. And so if there's a cysteine in this part of the protein and a cysteine in this part of the protein, they can then bind together, forming a disulfide bond. And this then links this portion of the protein to another. And so this helps to then form this tertiary structure. I had mentioned that some amino acids are acidic, some are basic. Depending on the pH, then they could have a charge. And so one could have a positive charge, one could have a negative charge. And therefore, the positive charge could then be attracted to the negative charge, forming what's called a salt bridge. So charged amino acids, acidic and basic amino acids, could then change the tertiary structure. So these parts of the protein could then be linked, all right, because of a salt bridge. And so in addition to just coding for an amino acid order, proteins have to form correctly. And this is something we can talk about later because that's a complex process. Folding proteins correctly, there are chaperone proteins which help to fold proteins correctly. Very often this has to occur in a specific part of the cell, like the RER, where it can be more acidic, otherwise the disulfide bonds don't form. And so, how do we correctly fold a protein to make it look like this. That can be an involved process. Finally, once you have a protein, sometimes that protein then meets with others. So a protein coded by one gene could then be incorporated with proteins made by other genes to form kind of a complex. And this is what is known as the quaternary structure of the protein. So the collagen that I had used as an example, there were different collagen genes. Each produced a polypeptide chain, but the protein collagen that you find in a cell is now the product of the pink collagen protein made by one gene, the aqua collagen protein made by a different gene on a different chromosome, etc. So how do different proteins then coalesce into a larger structure that could be determined as the quaternary structure? Not every protein does form these complexes. Sometimes the complex can be formed by more of the same. So if this is a p53 protein, this is a tumor suppressor, we could then take 4 of them and put them together. Or tyrosine kinases, we could put 2 of them together. And so you could have multiple subunits coded by the same gene or coded by different genes. So for example, hemoglobin in the blood, there are 2 alpha hemoglobin proteins coded by the alpha globin gene, 2 beta hemoglobin proteins coded by the beta hemoglobin gene. And this then forms hemoglobin. The quaternary structure of hemoglobin is then how these different subunits then coalesce. Okay, so once again, there's a number of things that just have to be said about proteins, about the genetic code before we can discuss translation. Moving on to the next topic, we have to discuss tRNAs. So tRNAs would be an example of an area which is encoded by DNA, which is converted into RNA, but that the RNA will never be converted into protein. So here you see RNA, and this is the final product, the RNA, which we call transfer RNA, tRNA. This is not an intermediary meant to code for a protein, but rather this is the final product. Now, tRNAs, they form these small structures with these 3 loops. Now, I should stress that this is a nice way to draw it, but this is assuming that they lay flat. They don't lay flat, so in 3 dimensions, they don't look like this, but this is, convenient for our conversation. So tRNAs are these little tRNAs which will be essential in the process of translation, so we need to discuss them. Now, they have these weird shapes with these loops. And if you ask, why do they have those loops? The answer is when we introduce the concept of RNA and we say, oh, there's 4 bases. There's adenine, guanine, cytosine, and uracil. That's true as a starting point, but these can then be modified. There are lots of different modifications of RNA bases. And so one of the reasons we used to think that RNA was boring and protein did everything is because the 20 side chains of the amino acids, they have things that they can do. But RNA nucleotides, they can be modified to form a fair diversity of structures, which can have many of the same roles that then, say, the amino acid side chains could have. So RNA is more diverse than others. And so if you look at the tRNAs, one of the things which gives it the odd shapes is that where there was once guanine, adenine, cytosine, and uracil, these have then been converted to modified RNA bases which don't pair with others. So notice that here, these structures, this sequence could be complementary to that sequence. That's why they pair up like that. Whereas here, there are modified bases which would prevent that that pairing. Some modified bases are common to all tRNAs. Some define specific tRNAs. So for example, lysine bases are characteristic of the tRNAs for phenylalanine. And so one of the players in translation will be these tRNAs, which have this unique shape because of many of the modified bases. Now, the genetic code is nice to put on a piece of paper and say, here's the code, that this codon, signifies this amino acid. But let's face it, that's a nice thing in the piece of paper. Where does the genetic code exist in a cell? So in a cell, where is the genetic code real? And the answer is in the tRNAs. So the tRNAs and in the enzyme which charges them, that's where the genetic code lives. And so what we want is that there is a nucleotide sequence, all right, in the DNA, which is copied into RNA. We want there to be a nucleotide sequence which has meaning. So for example, this mRNA codon, adenine, uracil, guanine, AUG, we want that to have meaning so that a cell says, whenever I encounter this codon, that means something. That means I'm going to put a methionine in that protein. So how do we do that? Well, these transfer RNAs, they're, in some ways translators speaking two different languages. They speak both the language of the RNA nucleotides, because they are made of RNA nucleotides, and they can have an anticodon, which is complementary to the codon. But they are also able to bind specific amino acids, speaking the language of proteins. Proteins are made of amino acids. So here we have a molecule that's going to do RNA speak and amino acid speak, and thus be able to link this RNA word to this amino acid, and thus the amino acid order of proteins. Now, there are different tRNAs, because one tRNA is going to need to bring methionine to any word that says put a methionine here. So this tRNA is what we call the tRNA MET for methionine. And so there is an enzyme which recognizes tRNA MET and will then take the floating amino acids and attach them to the other end of the tRNAs. So tRNAs have two vital ends. They have this end, which will attach the amino acid, and this end, which serves as the anticodon. And an enzyme recognizes this tRNA. It's methionine tRNA synthetase, and it will then take the amino acid and stick it here. So once again, a translator needs to speak two languages, all right? Well, here, this tRNA is going to speak both RNA speak with the anticodon. Once there's an amino acid attached to it, it now speaks protein as well, all right? And now it can take this part of a protein to any word that says we need that amino acid in this spot of the protein sequence. Once methionine is attached, the tRNA is what's referred to as charged, all right? So this is a charged tRNA MET. It has methionine attached. And so in the process of translation, which will be the topic of the next video, this tRNA can then go to any place where there is an mRNA codon in the messenger RNA. And here, there is an anticodon, all right? The RNA nucleotides are complementary. And so therefore, the anticodon UAC is complementary to AUG, with uracil complementary to adenine, adenine complementary to uracil, and cytosine complementary to guanine. So if in protein construction, then a methionine is required, this tRNA can bring methionine to the required spot. And then the same will go on because there are more amino acids. Valine is going to be required at some parts of the protein. So we need a tRNA for valine, so that anytime there's an mRNA codon that says we need a valine here, valine can be brought to that spot. And so the tRNA for valine begins uncharged, all right? So this tRNA for valine is not charged yet, but there is an enzyme which will specifically recognize the shape of this tRNA and attach valine to it. So this is valyl tRNA synthetase, and it will then take valine and charge the tRNA. Once this tRNA is charged, tRNAs can then approach the mRNAs. And whenever there is an mRNA codon, which specifies valine needs to be inserted here in the protein structure, that then valine can, in fact, be inserted there. Okay. And this is because then there are, once again, two important parts of the tRNA, not only the side that binds the amino acid valine, but here the anticodon, all right, which will then be complementary to the mRNA codon. If the mRNA codon reads GUG, then this anticodon is complementary CAC. And so forth. Just one more quick example. This would then be true for every amino acid. Every amino acid has to have a corresponding tRNA, and sometimes there's more than one kind of tRNA, which we'll get to in a second. So alanine needs to be brought to mRNA codons, which say put an alanine here. And so there are tRNAs for alanine. There are tRNA synthetases, which will then recognize this tRNA and attach an alanine to it, at which point then the tRNA is then charged, and then whenever this anticodon recognizes the mRNA codon, it will bring alanine to a specific spot. So tRNAs are translators, essentially speaking two languages. Now, at this point, we can discuss the genetic code in general. The first thing about the genetic code is that it is a triplet code. So when we look at the 3 billion nucleotides, many of which then get transcribed into RNA nucleotides and transcripts, there's meaning there. And some of the meaning is put this amino acid at this place. So some sets of RNA nucleotides, they are essentially words of a language. Okay. Now, if a codon, if a word was composed of a single nucleotide, so if uracil was a word that meant something, if cytosine was a word that meant something, if adenine was a word that meant something, then because there's only 4 nucleotides, we would then only have 4 words. And so proteins could then be made, say, of 4 amino acids, but no more. Now, because there are 20 amino acids, then this is clearly not enough. If words were composed of 2 nucleotides, so if UU meant something, if UC meant something, UA, UG, then there would be 16 possible words. Now, since there are 20 amino acids, this then is a problem. That would not be enough. All right, so we need more than 16 words to code for 20 amino acids. So a codon wouldn't be able to have 2 nucleotides in it. And so the smallest length, which would then allow us to code for all 20 amino acids, would be if it is a triplet code, where there is the first nucleotide is either G, A, C, or U. So notice that here with U, all of these, the first nucleotide is U. Here, all of them, the first nucleotide is C, etc. The second nucleotide is either U, C, A, or G. So notice these all begin with U, and then the second nucleotide is U. Here, the second nucleotide is C, the second nucleotide is A, the second nucleotide is G. And then the third nucleotide is either U, C, A, or G. So there's U, C, A, or G. So the DNA code is a triplet code, so that the words, which have meaning, then have three different codons in them. Now, as we describe this code, the term that's typically used is degenerate. I don't think that's the best term necessarily. The term degenerate here means that the genetic code has synonyms, all right? So, I would argue there's a better way perhaps of saying that there are synonyms here. But for example, if you wanted to say, put an alanine here, if that's an instruction that you wanted to give to, the ribosomes making a protein, how do you say put an alanine here? Well, GCU is a word which says put alanine here. GCC is a word that says put alanine here, GCA, GCG. Why? Well, because there are 20 amino acids, and if there are 4 possibilities for the first nucleotide, times 4 for the second nucleotide, times 4 times 4 times 4 is 64. There are 64 combinations, but only 20 amino acids. So then obviously, it's not going to be a one-to-one correlation. And so here, there are 4 different codons which code for the same amino acid alanine. There are 2 codons which code for aspartic acid, and 2 which code for glutamic acid. There are 4 which code for glycine. There are 6 which code for leucine. That's the only example where there are more than 6 coding for leucine. And so as we describe the genetic code, we use the term degenerate because there are synonyms. So there are multiple codons which mean the same thing. Now, because of that, and I'll get into WOBBLE in just a second, sometimes this third codon isn't important. So if GCU, GCC, GCA, and GCG all code for alanine, then all you really have to recognize is the GC, all right? Because no matter what the third one is, you're going to code for alanine. Here, no matter what the third one is, you code for threonine. Here, no matter what the third one is, you code for proline. Here, so the third codon is not as vital in transferring meaning. There are 3 cases where you absolutely have to know the third, I'm sorry, when I say third codon, I mean third nucleotide in the codon. So AUG means both methionine and start, and you have to know that G because no other codon codes for that. UGG codes for tryptophan, and UGA codes for stop, okay? But in all other cases, it is possible then that you could have the exact same meaning with a different third position in the codon. Cytosine and uracil are never meaningful in the third position because GCG is the same as GCU. UGC is the same as UGU. And so when we talk about translation, adenine in the third position is only meaningful when it codes for stop. Now, there'll be other applications of this, I don't want to get into them here, but this will obviously be revisited when we talk about mutations. So this minimizes the effects of mutation. So if there's a mutation in the third position so that this U is converted to an A, it would still code for the same amino acid. So mutation, which is a later topic that could be brought up. Now, of the 64 possible codons, there are some examples of more or less punctuation. So methionine is coded for the codon AUG, but methionine is always used to start translation. The methionine is always the first amino acid in a protein, although it can later be removed. So it doesn't have to stay the first amino acid in the protein. And then there are 3 codons which do not have a matching amino acid. So they do not code for an amino acid. They then code for stop. So when you start making a protein, you begin with AUG. When you stop making a protein, you use one of these 3 stop codons, UAA, UAG, or UGA. Okay, so there is no amino acid which codes for that. So there's a bit of punctuation in the genetic code. One of the interesting things about the genetic code is that it is universal. So if you take a human gene and put it into a turtle, the cells of the turtle make the same protein as the human cells would. If you put a human gene into a plant, the plant would make the same proteins. They would read it the same way as we do. Now, this is significant, so forgive me, let me just stress this point. There are words, all right, that you can use in different languages. So just because this word means this in this language, the same word could be used in a different language and could have a different meaning, all right? Even different, people who speak English or Spanish, you could use this word in Colombia and it has a slightly different meaning in Argentina, or this word used in England has a different order than Australia. Sign language is the same way, perhaps, this is an easy one. So you can do this, all right? What does that mean? Well, it means, things are good, all right? But if you're scuba diving, it means things are bad. We need to surface right away. So you never do this if you're good in scuba diving, you do this. So here's an example where you can use the same symbol, all right? But it could mean different things in different languages or different cultures. So the same thing here, just because you is a word, and in humans it means put a phenylalanine there, that doesn't mean that a plant would have to use you, all right, to say put a phenylalanine there, right? They could say, use AGU. You could express meaning the same way, all right? People who speak Spanish, all right, ellos pueden usar palabras diferentes para el mismo significado. They can use different words to get the same meaning across. All right, so meaning doesn't have to be tied to these words or that words. A different language can use a different set of words. But in living organisms, we essentially all speak the same language. This is the evidence that the genetic code, in essence, evolved once, and that all living things alive today are descended from the first living things to use this genetic code, because our cells still speak the same language. If living things had different origins, one could expect wildly different codes, but even if there was the same code, the words could mean different things. So all life shares a common ancestry, and the common usage of the genetic code is one example of that. Now, even when you speak the same language, and forgive me, I really should learn better examples of this, but the first one that comes to mind is if you were to say, I'm pissed, what does that mean? Well, that depends. You're speaking English, but where are you speaking English? In the United States, that means you're angry. In England, it means you're drunk. All right, so the same word could, even though people are speaking English, almost all of their words are the same. You could have some regional variations. And so there are a few regional variations of the DNA. So while it is a universal language, there are a couple cases of accents where one organism uses a word that is different from the universal language. And so some paramecia read these stop codes as glutamine. One ciliate reads this stop code as cysteine. Some bacteria read this stop code as tryptophan. And interestingly, because mitochondria have a slightly different accent in the genetic code compared to eukaryotic cells, mitochondria, they are descended from bacteria. So inside your cells, you have mitochondria descended from bacteria. The genetic code of mitochondria speaks with the bacterial accent, not with the accent of the eukaryotic cell. Okay. A couple final quick points. Mutations often come, later in a genetics course in discussing these. But just, a couple quick definitions here, and these can then be repeated later. So, what is the effect of a mutation? Well, when we look at the genetic code, because there are synonyms, if you changed CCU to CCC, technically that wouldn't have an effect on a protein. Now, life is more nuanced than this, but this is a good starting point. The nuance can come later. So this would be what's called a silent mutation, in that it doesn't change the amino acid order of the protein. A nonsense mutation, by definition, is one that changes a coding amino acid to a stop codon. So lysine is coded by AAG, but if a mutation changes this first A to a U, so now that the code for a protein, instead of having an AAG, now has a UAG. That means there's a stop codon. That means that the resulting protein will be shorter than it otherwise was meant to be. Lysine was meant to be somewhere in the middle of the protein, but now it says stop. That codon's now the end of the protein. That's what's called a nonsense protein, and that can result in a truncated protein being shorter than it usually would. Okay, and so sometimes if you ask, why does this person's version of, say, BRCA1 lead to cancer? The answer is there is a nonsense mutation which is truncating the protein. It's too short, and therefore it doesn't work. Then finally, another class could be described as missense mutations, where one amino acid is now changed for another. Right? So what causes sickle cell anemia, for example? Well, the normal glutamic acid is now changed into valine. And that's significant because glutamic acid is an acidic amino acid. Valine is nonpolar. This has a big effect on the structure of the protein. So if you change a proline to a threonine, et cetera, then you have what's called a missense mutation. And once again, that's serious. That can be serious, especially if you're changing, say, a polar amino acid to a nonpolar, et cetera. If you change one nonpolar amino acid to another nonpolar amino acid, maybe it doesn't have that big of an effect on the protein, but that can be a topic for later. So understanding the genetic code is then also important in understanding, what possible effects of mutations could be. In translation, the next topic, you could mention the word wobble, that when the tRNA with its anticodon matches the codon of the mRNA, the first position has to match. The second position has to match. But remember how I said the third position often does not have meaning? The ribosome will often accept a little bit of wobble, where not only will this cytosine be allowed to complement that guanine. That's the appropriate. But another third position could be allowed to complement that last one in the codon, because once again, the third position doesn't necessarily have meaning. So as a result, if you ask how many tRNAs am I making in my cell, am I really making 61 all the time? Well, sometimes no. Sometimes you're making a smaller set because there's just a bit of wobble that one tRNA could actually match multiple mRNA codons. So UUG would not only match AAC, but it would also be allowed to match AAU. And therefore, you wouldn't have to make UUA, a different tRNA, because this one, the cell would accept to match two different mRNA codons. That's what's meant by wobble, that the pairing of the third position in the codon anticodon pair, there is a wobble accepted. It doesn't necessarily have to be exact in many cases. And if, in addition to guanines and cytosines, if one of those positions was then modified, once again, if we had one of those modified tRNA bases, then that would allow multiple different pairings. Okay, so in the central dogma, DNA is converted to RNA, which is converted to the amino acid order of a protein, and translation, where the RNA transcript is translated into protein, is the next topic. But prior to that, just the structure of proteins, the structure of tRNAs, and the aspects of the genetic code had to be mentioned. So that's what this video was meant to do.